All right, today we're going to crack a code. Not some computer code, but a secret language hidden deep inside your DNA. It's the language of your immune system. It's called HLA typing. And, understanding it is absolutely critical. So let's just dive right in.
So take a look at this. At first glance, you're probably thinking it looks like a software version number or maybe a Wi-Fi password you'd never remember. But what if I told you that this string of letters and numbers is actually a fundamental piece of your biological identity, written directly into your genes?
So what's the big deal? What does this secret code actually mean for you, for your health? And why is it one of the most important codes in your entire body, playing a role in everything from fighting off a cold to accepting an organ transplant? Well, that's exactly what we're here to figure out.
So here's how we're going to tackle this. We'll start with this mysterious genetic code. Then we'll unpack the body's incredible self-ID system. After that, we'll take a look at the history of how the technology got better and better at reading it. Then comes the fun part. We'll crack the naming code itself, explore the different levels of detail or precision, and finally, we'll decode some of the weird special symbols you might see in a report.
Okay, so let's start with the puzzle itself. Before you can crack any code, you've got to understand the system it belongs to. And for us, that system is the Human Leukocyte Antigen system, or as you'll hear it called, HLA.
The easiest way to think about the HLA system is to imagine it as your body's own internal immune ID card. It's this group of genes on chromosome 6 that make special proteins for your cells. These proteins are constantly showing little pieces of what's going on inside the cell to your immune system, basically saying, hey, I'm one of the good guys. I'm self, I belong here. But if a cell shows the wrong ID, or maybe an ID that's been hijacked by a virus, well, that's when all the alarm bells go off. And this system is, it's unbelievably diverse. We're talking over 28,000 known versions, or alleles, as scientists call them, of these genes. And that's no accident. It's actually a brilliant evolutionary strategy. The more variety our immune ID cards have across the whole human population, the better we are as a species at fighting off a massive range of different viruses and bacteria. Now, it's not just one thing. There are two main types of these HLA molecules. Class 1 molecules are on pretty much every single cell in your body. Think of them as sending out a constant internal status report. They show patrolling immune cells little bits of what's happening inside the cell. If a virus gets in, parts of that virus get displayed, and the immune system knows to take that cell out. Class 2 molecules are different. They're only on specialized immune cells, and they act more like wanted posters. They show off fragments of invaders they've found outside the cells, like bacteria, to rally the rest of the immune system for a big, targeted attack. So how in the world did we get from just knowing the system exists to being able to read that super complex code we started with? It's really a story about technology, and you can think of it like a camera's resolution getting sharper and sharper over the decades. Way back in the day, we had something called serology. This was like a really blurry, out-of-focus photo. It could tell us the big, broad groups, but that was about it. Then in the 90s, things got a bit clearer with PCR-based methods, kind of like switching from an old tube TV to standard definition. The 2000s gave us Sanger sequencing, which was the gold standard for a really long time. Now we're in high definition. But today, with something called next-generation sequencing, or NGS, it's like we're watching the whole thing in stunning 4K video. We can see the entire gene, every little detail. And here's what's really interesting. There's a trade-off with each of these methods. Serology was super fast and cheap, but the resolution was terrible. Sanger is incredibly accurate, but boy, is it slow and expensive for just one sample. And NGS, well, it gives us the most complete picture by far, but the data analysis is incredibly complex and the machines cost a fortune. So choosing the right tool always depends on the job you're trying to do. Okay, so now that we've got the backstory on the system and the technology that reads it, it's time. Let's finally go back to that original code and crack this thing once and for all. All right, here it is again. Let's actually tear it apart piece by piece. So you see those colons, right? Each part of the name separated by a colon is what we call a field, and each one adds another layer of detail. That first field, the 01, tells us the broad family, or the serological group. Think of it like a last name. In this case, it belongs to the A1 family, something we could spot even with that old, blurry technology. Now the second field, the O2, zooms in a lot closer. This tells us the exact protein variant that's being made. A tiny, tiny change in the DNA code can alter the protein, and that difference is everything when you're trying to do something like perfectly match an organ for a transplant. Okay, this third field is really cool. It identifies what's called a synonymous mutation. This is a quirk of genetics where the DNA recipe changes, but the final dish, the protein, comes out tasting exactly the same. It's like spelling the word color versus color. Different spelling, same exact meaning. So why do we even care? Well, for researchers, this is a huge clue for tracing how genes are passed down through a family.
And that brings us to the fourth and final field. This one points to changes in the non-coding parts of the gene. These are bits of DNA that don't actually build the protein itself, but they can act like a dimmer switch, controlling how much of the protein gets made. A change here might mean a weaker immune signal, which could have big implications for disease risk. So, as you can tell, we can get ridiculously specific. But the thing is, you don't always need all four fields. The right level of detail, the right resolution, really depends on the question you're asking. You can think about it like a pyramid of precision. At the very bottom, you have low resolution, using just that first field. It's great for broad population screening. Move up a level, and you get high resolution, which adds the second field to identify the specific protein. That's a game changer for medicine. Add the third field, and you get allele resolution, which is great for tracing family genetics. And right at the very top, you have the full sequence. That's the whole shebang, the most complete picture you can possibly get. We're almost there, but there's one last thing you need to know. Sometimes you'll see a weird letter or a symbol tacked on to the end of an HLA type. You absolutely cannot ignore these. Think of them as critical footnotes that give you the real context of the result. These letters are basically little warning flags for the scientists. If you see an N, it means that allele is null. It's a dud. It doesn't function. An L means it's working, but it's on low power. It has low expression. A Q flags the result as questionable and means you better double check it. And symbols like a slash or a plus sign tell you that the test couldn't land on a single clear answer. You can see how missing one of these little letters could totally change the story. So there you have it. We've gone from a completely cryptic line of text to a detailed, meaningful biological address. This code is the key to matching organ donors, to predicting our risk for certain diseases, to designing new medicines. It's a fundamental piece of who we are. And it really makes you wonder, doesn't it? Now that we've cracked this incredibly complex code, what other biological languages are we on the verge of finally understanding next?